Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get into med school and other professional programs. Today, we're going to talk about nucleic acids and how they replicate, acquire mutations, as well as undergo repair. All important things you'll be expected to know for the MCAT. Nucleic acids are the molecules that store and transmit genetic information. They're composed of nucleotides, which are made up of a sugar, a phosphate group, and a nitrogenous base. There are two types of nucleic acids, DNA and RNA. DNA is the primary genetic material in cells and carries the instructions for the development of functions of all organisms. First, let's look at how DNA is replicated. DNA replication is the process by which cells make a copy of their DNA before cell division, often during S phase. The replication process ensures that each new cell receives a complete set of genetic instructions. Replication begins at a specific site on the DNA molecule known as the origin of replication. Enzymes, called helicases, unwind the double-stranded DNA molecule, creating a replication fork. DNA polymerase enzyme then joins the party and adds nucleotides to each of the two separated strands of DNA, creating two new strands of DNA that are complementary to the original strands. DNA replication is a highly accurate process with an error rate of less than 1 mistake per billion nucleotides. Despite the accuracy of DNA replication, mutations can occur. A mutation is any change in the DNA sequence of an organism. Mutations can arise spontaneously as a result of errors in DNA replication or repair, or they can be induced by external factors such as radiation or chemicals. Mutations can have a variety of effects on an organism. Some mutations can have no effect at all, while others can have severe consequences. Mutations in certain germ cells can lead to the development of diseases such as cancer, cystic fibrosis, and sickle cell anemia. One type of mutation is known as a point mutation, which involves a change in a single nucleotide in the DNA sequence. These three, silent, nonsense, and missense, are all different types of point mutations. Silent mutations do not affect the amino acid sequence of the protein being produced. If we look up here comparing to the normal, so we're saying that the cysteine is turning into a T. Well, thanks to redundancy in the codon sequence, we're still coding for alanine. So this is a silent mutation, a mutation that changed the code of DNA, but it's having no protein effects, so it really doesn't matter. Next up, we have nonsense mutations. Nonsense mutations result in causing a stop codon too early on. So this is going to drastically shorten the protein being encoded, which is usually going to have very negative effects. Next up, we have a missense mutation. Missense mutations result in a different amino acid being coded for. So originally we had a cysteine being coded for up here, but now we've taken the thymine, turned it into guanine, and now we are making an arginine for that third amino acid. Who knows how drastic of a problem this will be, but it is a change. The next type of mutation we have is called a frameshift mutation, and all three of these are frameshift mutations. These involve the insertion or the deletion of amino acids, insertion or deletion of nucleotides in the DNA sequence. Frameshift mutations can have a significant impact on the resulting protein sequence, as they shift the reading frame and can result in the incorporation of multiple wrong amino acids. Normally, frameshift mutations are worse than silent and missense mutations. When we have a mistake in replication or in mutation, the first step is to try and repair it. One common mechanism that we try and repair DNA is known as base excision repair, or BER. This process begins with the removal of a single damaged DNA base by a specialized enzyme. The resulting site is then recognized by other enzymes which remove the damaged DNA and replace it with a new undamaged strand. You'll notice I'm being very vague because you don't need to know the name of these enzymes, so I'm not going to clutter your brain with information you don't need. The other term you should be familiar with DNA repair is known as nucleotide excision repair, or NER. This removes a large chunk of damaged DNA and replaces it with new undamaged DNA. And finally, you have to know about MMR, or mismatch repair. All you need to know for the MCAT about this one is that it occurs during G2 and helps repair damaged DNA. So this one is even more vague than the other two. Isn't that nice? DNA replication, repair, and mutations are all critical processes that ensure the accurate transmission of genetic information and the maintenance of the integrity of the genetic material. While mutations can have significant consequences, we didn't really talk about how they do have a positive role in promoting genetic diversity. Even if it is a bad mutation 99% of the time, that 1% leads to the development of new species and improvement, which is essential for the evolution of species over time. You should now feel comfortable with these topics when tackling them on the biochem or chemistry sections of the MCAT. Thank you so much for watching our video, and I will see you next time.